Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today's topic is DNA modifying enzymes. In this session, you will be learning about what are the DNA modifying enzymes and what is the purpose of those enzymes in recombinant DNA technology, different types of enzymes, the mechanism of action and the application of these enzymes in gene cloning experiments. Dear learners, let's study about DNA modifying enzymes in detail. So these enzymes, they affect the change in the DNA chemical constitution by adding certain functional groups or removing the groups like phosphate group or hydroxyl group. In that way, they will be changing this chemical constitution of the DNA, and also by means of gain or loss of the bonds, the changes are made. So the enzymes which change the chemical constitution either by addition or removal of the functional groups or by the net gain or loss of covalent bonds, those are called as DNA modifying enzymes. So these DNA modifying enzymes are of different types. The one which we will be studying in this session, they include exonucleases, polymerases, phosphatases, kinases, terminal transferases and ligases. Coming to the first type of DNA modifying enzyme, exonucleases. As the name suggests, the nucleases are the one which cuts the nucleic acid, and exo means they cut either the 5' end or the 3' end or both the ends. So the exonucleases which are used in gene cloning experiments, they are extracted from a marine bacterium called Altermonas aspergillana. It is called as BAL31 nuclease, and the other one is E. coli. The enzyme is exonuclease 3. So this exonuclease, that is BAL31 nuclease, is having the ability to cut the DNA from both the ends, that is in the 3' and 5' end from both the sides. Suppose you incubate the DNA fragments with this BAL31 exonuclease for a period of time. As you increase the time period, the length of the fragment shortens. So in that manner, the exonuclease cuts both the strands and it shortens the length of the DNA. The exonuclease 3, it cuts particularly at the 3' ends. So 3' ends are cleaved, and thereby leaving the 5' strand, single stranded 5' regions are present. Overhangs, 5' overhangs will be left over. So whenever you treat the DNA sample with this exonuclease 3, you will be getting DNA fragments having 5' overhangs. So in this manner, to shorten the DNA fragment or to produce overhangs, these particular exonucleases are used. Coming to the second one, DNA polymerases. So these enzymes, they catalyze the synthesis of new strand from the pre-existing strand. During replication, you will be seeing this thing, and also during chain extension. So the enzymes, they add the DNTPs to the free 3' hydroxyl group, and thereby the chain will be extending from 5' to 3' direction. The overall reaction is represented here. The DNA is the polymer of this DNTPs. So each nucleotide is added by the removal of this pyrophosphate. The major requirements for this polymerase activity, they include first a template strand, a primer group, and this building blocks, that is deoxyribose nucleotide triphosphates. The 4 nitrogen bases you will be requiring. So the polymerases which are used in recombinant technology, they are of three types. One is proofreading polymerases, thermostable DNA polymerases, and reverse transcriptase polymerase. So whatever may be the type, the basic mechanism of action is it will be same in all the this type of polymerase enzyme. So this is the pre-existing strand. It's a polymer which is containing two nucleotides, and if you are adding a monomeric unit, that is the DNTP is added to this one, this one is monomer is added to this particular polymer by the formation of phosphodiester bond. This phosphodiester bond is formed between 5' carbon of this, that is the 5' carbon is this alpha phosphate is reacting with the hydroxyl group of the which is present in the 3' carbon of the existing strand. So by the formation of the phosphodiester bond between the 5' carbon of this monomeric unit and the 3' hydroxyl group of this strand, you are seeing the formation of the phosphodiester bond, and during this process this pyrophosphate it is removed. So in this manner the basic reaction it is same in all types of polymerases. So the three types of polymerases which are used are proofreading polymerases. So the that apart from the main function polymerase activity, they are also having this 5' to 3' exonuclease activity, then 3' to 5' exonuclease activity, that is proofreading properties, additional property present in these particular type of polymerases, and these polymerases are used in this PCR reaction, mainly applied they are used in the application of this PCR. And the proofreading polymerases are generally they are extracted from Pyrococcus, then this Thermococcus species. Then the second one is TAC polymerase. This is the thermostable DNA polymerase. So they require DNA as a template, and they require DNTPs and magnesium ions. Apart from the polymerase action, it is having the additional activities this 5' to 3' exonuclease, and this enzyme is also used in PCR. The third type is reverse transcriptase enzyme. So here the template taken here is RNA. So in the previous one you will be seeing DNA as the template, but in this one the RNA will be is acting as a template, and you will be having the primer. The enzyme it is having reverse transcriptase activity, and apart from that it's an RNA dependent DNA polymerase activity, and it's having a additional function is this ribonuclease activity is also there. So this reverse transcriptase is used in RT-PCR experiments. So all the examples we will be learning this one in particular, reverse transcriptase. So this is an RNA dependent DNA polymerase. This enzyme is extracted from retrovirus, and here the reverse transcriptase takes this RNA molecule as a template, as a base for the synthesis of the DNA. So for this purpose it's called as RNA dependent DNA polymerase. So with the help of the primer, by utilizing this RNA as a template, from this if you add the monomeric units, that is the DNTPs, from this the chain is extended by means of the polymerase reaction. So at the end of the reaction you will be getting RNA and DNA hybrid. This hybrid, even it is treated with this ribonuclease. Ribonuclease activity is also present internally in this reverse transcriptase enzyme. This RNase H activity, it cleaves the RNA strand from this particular hybrid, and it leaves the single stranded DNA. This is called as complementary DNA. So this complementary DNA, it is exclusively it is used for cloning in expression vector and in the production of the this transgenic organisms, cDNA plays a very important role, and it is also helping in this cDNA library construction also. Coming to the next class of enzymes, phosphatases. So phosphatases, they catalyze the cleavage of phosphate group from the 5' end of the DNA molecule. You know that the DNA contains phosphate group at the 5' end and hydroxyl group at the 3' end. That 5' phosphate is removed by these phosphatases, and this reaction is reversible. So the phosphatases which are used in gene cloning experiments, they are extracted from the calf intestine. So it's called as calf intestinal alkaline phosphatase, and this particular enzyme is used to cleave the phosphate group from the 5' end of this DNA as well as RNA. And why these phosphatases are used means in order to prevent this self ligation of the vector molecules. So whenever the vector molecules or the target DNA, they are having this cohesive ends, they are having the ability to again show this self annealing or recircularization is will be happening. In order to avoid that, this particular phosphate group is added, it is modified with the hydroxyl group. So when this hydroxyl group is present at this ends, it failed to anneal. So in order to prevent that self annealing and recircularization, this particular modification is done by means of alkaline phosphatase. So in this example, the 5' phosphate is converted to this 5' hydroxyl group. The same is represented here. It is present in the phosphate group is present in this 5' carbon atom. That one is modified with this hydroxyl group in the presence of this alkaline phosphatase. The next enzyme is polynucleotide kinase. This enzyme works in an exactly reverse type as that of the phosphatase. So kinases, they add phosphate groups, whereas you have seen that phosphatases remove the phosphate group. These kinases, they add the phosphate group to the this 5' end. So after the reaction, that is when you are ready for inserting the target DNA into the vector, so in order to insert that target DNA, first the hydroxyl group which is present in the 5' end, it should be removed. So that one is done by using this polynucleotide kinase. So it helps in the transfer of phosphate group. For this ATP is required. So the phosphate group present in the ATP, it is transferred to this 5' end by means of the enzyme polynucleotide kinase. So these polynucleotide kinases, they are extracted from T4 bacteriophage. So that is same thing is represented here. So the gamma phosphate group of this ATP, it is transferred to the terminal 5' end of this particular DNA strand, and thereby modifying the hydroxyl group to phosphate group. The next enzyme is terminal transferases. This enzyme is extracted from calf thymus, and it helps in the addition of this nitrogen bases or this deoxyribonucleotides DNTPs to the 3' terminus of this both the double stranded and the single stranded DNA. So particularly this enzyme, it is used for labeling the 3' ends of the DNA. Since you are adding same type of this bases, that is the DNTPs at the 3' ends, these are called as homopolymeric tails. So in the plasmid, in the vector DNA and in the target DNA, you are adding a complementary type of nitrogen bases. Here see in this plasmid you are adding GGG, homopolymer tail, the short fragment of DNA containing this nitrogen base is G, whereas in this particular target DNA you are adding DCTP. So the 3' end of this target DNA you are adding a homopolymer tail of DCTP, whereas in the 3' end of the plasmid you are adding homopolymer tail of DGTP. So if you are adding this type of complementary homopolymer tails to two different DNA molecules, that is target DNA and vector DNA, you can easily seal them, because due to transient base pairing you can first easily they will be undergoing hybridization, and later the discontinuities, the gaps, they are sealed by means of this DNA ligase. So these terminal transferases are used to insert the target DNA into the vector molecule. Then the last one is DNA ligase. So this helps in joining the DNA fragments by the formation of the phosphodiester bond between 3' hydroxyl and 5' phosphate group present in the this particular double stranded DNA, and they require high energy phosphate groups like this, that is ATP and NAD is required for this reaction. And the DNA strand, suppose you want to join these two DNA strands, so the bond is formed between the 3' hydroxyl group will be attacking the 5' phosphate group of the adjacent strand. By the formation of this phosphodiester bond, the two strands, they are joined. For this ligation you require ATP and NAD plus as the cofactors, and it's a energy consuming reaction. And ligation, by means of the ligation you can join different types of DNA belonging to different origin. So coming to the conclusion, the DNA modifying enzymes which we have studied, they modify the chemical constitution of the DNA by addition or removal of the functional groups, or by gain or loss of the covalent bonds. The exonucleases, they cut the double stranded DNA from the 5' end or the 3' end or both the ends. The polymerases which we have studied, it adds DNTPs to the free 3' hydroxyl group of the chain, thereby you can extend the chain. And the phosphatases, they remove the phosphate group from the 5' end. And the kinases, they add the phosphate group to the 3' end. The terminal transferases, they help in the addition of one or more DNTPs to the 3' end of the DNA, or they add homopolymer tails. And ligases, in the last, they will be helping in joining the DNA fragments by the formation of the phosphodiester bond. With this, I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.